Today I'm talking to you about standard ways of collecting microbiological samples from epiphytes. I'm going to start by defining what epiphytes are. Epiphytes are organisms that grow on the surface of plants without deriving nutrients from them. Epiphytes are different from endophytes. Endophytes live inside of the plant, but epiphytes live on the surface of the plants and they do not derive nutrients from the plant. In essence, we can say that epiphytes are commensals on the surface of plants. We can also say that these epiphytes they come from the environment because they are dropped one way or the other on the plant from the environment. And epiphytes includes bacteria, fungi, and algae that reside on leaves, on stems, on flowers, and fruits. Studying epiphytic microorganisms is important for agriculture, for plant microbe interactions, for disease control, and for research and studies in microbiology and biotechnology. Proper microbiological sample collection ensures accurate analysis while minimizing contamination. This webinar today and this video outlines the standard method for collecting microbiological samples from epiphytes, including equipment, technique, and best practices. The first thing we'll look at is preparing for sample collection. And I'm going to tell you some of the equipment you need for the sample collection. So the equipment and materials needed to collect epiphytic microorganisms, you will need sterile swabs, that is cotton, synthetic or foam. Then you also need sterile faucets and scalpels. You will also need sterile sampling bags, e.g. wild pack or tubes. You can use test tubes that are sterile. You also need buffered solutions, e.g. phosphate buffered saline, that is PBS. Then you need sterile gloves to prevent contamination. You also need alcohol wipes, 70% ethanol or isopropanol. I prefer ethanol for sterilizing the tools you will use during sample collection. You also need ice packs and cooler for transporting samples from the field to the laboratory. Then you also need field logbook for recording the site details. Many students go or many researchers go around without a logbook, without a lab note. So the lab note helps you to put down everything you need to put down because the faintest pen, it is said, is sharper than the best memory. So you also then the site and sample selection. Choose LD and diverse plant species to get a broad representation of epiphytic microbes. Avoid areas exposed to excessive pesticides, pollution, or direct human handling. So if you are going to take from a field, then you have to look at the inner part of the field, not on the outer surface of the field. So sample different plant surfaces, including upper and lower leaf surfaces, stems, and flower petals. Collect at different times of the day or different seasons for microbial diversity studies. So don't just collect in the morning. If you are interested in microbial diversity study, you should be interested in microbial diversity study because it's going to add to the data you are going to generate. So you take sample in the morning, then you also take in the afternoon to compare what happens in the morning before the UV light of sunlight, and then what happens in the afternoon to the epiphytes when the UV light is up. So you can also collect at different seasons, maybe rainy season and dry season in Nigeria, maybe in summer and winter in some other places. Then in some other places you can collect in the summer, winter, and in the monsoon. Now, let's look at the standard method for collecting epiphytic microorganisms. The first one is the swabbing method, and it is for targeted microbial sampling. This method is ideal for collecting epiphytic bacteria, fungi and yeast from plant surfaces. What then is the procedure for this swabbing method? Wear sterile gloves and avoid touching the sample area directly. Moisten a sterile swab with sterile buffered solution, e.g. PBS or Peptone water if needed. Rub the swab firmly but gently over the plant surface in a zigzag motion. Immediately place the swab into a sterile collection tube containing buffer or transport medium. Label the tube and store it at 4 degrees C in a cooler until processing. The next thing you need to do is to rinse. You can rinse or wash. You can use rinsing or washing method. This is another method. The first method, as I said earlier, is the swabbing method. So majorly, most people use the swabbing method, but that is not the only method. Another method is the rinsing or washing method for collecting a broad microbial population. This method is used when sampling a large microbial community from plant surfaces. The procedure are as follows. Using sterile forceps, place leaf, stem, or flower samples into a sterile container. Add sterile buffer solution 10 to 20 milliliter to the container. Then gently shake or vortex the container for 1 to 5 minutes to detach microorganisms. Transfer the wash solution into a sterile tube and store on ice. So that is how you do that. The next method is direct contact plate method. This is for cultivable microorganisms. Remember that most microorganisms are not cultivable, are not culturable on nutrient media or on agar media. So this direct contact plate method is for cultivable microorganisms. This method involves pressing agar plates directly onto plant surfaces. So the procedure is as follows. Prepare nutrient agar for bacteria and pollutant stress agar for fungi. Press the agar surface onto the plant material for 5 to 10 seconds. Incubate at 30 degrees to 37 degrees Celsius for bacteria and 25 degrees Celsius for fungi. The bacteria will be incubated for 18 to 36 hours while the fungus will be for 1 to 3 to 5 days. Observe microbial growth and isolate colonies for further study. Then let's talk about the sample handling and transportation. Store all collected samples on ice, that is at 4 degrees C, and process them within 24 hours. Record sample location, plant species, environmental conditions, and time of collection in the field logbook. Avoid excessive exposure to heat or light, which may alter microbial populations. Now, let's look at the microbiological analysis of epiphytes. Once samples reach the lab, microbiological tests include culture-based methods, that is tricking on selective media for bacteria and fungal isolation. We also do microscopy, that is gram staining, lactophenol cotton blue staining for fungal identification. Then molecular techniques such as PCR, metagenomics for microbial diversity studies, and for confirmatory tests or identification of the isolates. Then enzyme activity tests to assess functional roles of epiphytes in plants health can also be carried out. In my own case, I carried out biodegradation tests, checking for the ability of these epiphytes and even sometimes endophytes to be able to produce biosurfactants that will help them to degrade petroleum. Collecting microbiological samples from epiphytes requires careful selection of plant materials and adherence to sterile techniques. Whether using swabbing, rinsing, or direct plating, researchers must ensure proper handling, storage, and timely analysis. These methods help in studying plant microbe interactions, disease control, and agricultural microbiology. So this is our webinar today on how you can do and correctly take sample from epiphytes. So if you have any questions or comments, you can drop it in the comment section below and I will come there to respond to you. And please subscribe to this channel. Please like this video. Please share this video with your family and friends. My name is Dr. Olubenga Adetitun, and this is our webinar series in the Environmental Toxicology and Petroleum Microbiology Research Group of the Department of Microbiology, Faculty of Life Sciences, University of Ilorin. I will talk to you in the next one. Please share the link of this video and like this video. This video popping up by the side from this channel is the next you should go ahead and see now. See you in the next one. Bye for now.